Dikeakos. Thank you so much for joining us. Today we'll be asking you some questions to better understand the courses you teach, the investigations you conduct, and the thesis projects you supervise for fourth-year honors specialization students. First, could you tell us a bit about yourself?
Great, I'm really happy to join in this interview. I'm Jimmy Dikeakos. I'm an associate professor in the Department of Microbiology and Immunology at Western University in the Schulich School of Medicine and Dentistry. And I would like to consider myself a virologist, so somebody who primarily studies viruses.
Okay, that's great. Can you briefly tell us a bit about what you're investigating?
Sure. Our lab is really interested about how things move around in cells, so primarily we're interested in membrane trafficking. So fundamentally, how different proteins will go to different locations inside of cell. What we're really interested in with that respect is how viruses such as HIV, which is human immunodeficiency virus, how they can basically attach to different proteins that are human proteins and change their location. So we look at a lot of this dynamic nature to see how the protein movement of human proteins will be affected by viral proteins that are expressed by the HIV virus.
Okay. And could you also explain to us what this lovely machine here is about?
This is basically a thermocycler. So we basically conduct a very basic but fundamental experiment in the lab, which is called the PCR, or polymerase chain reaction. So basically what this can do is quite simply change different temperatures and allow small pieces of DNA called oligomers, right? They can bind to various locations within a plasmid DNA and amplify different regions and make them bigger and insert these different sites. So essentially what we use this for is to clone. So we can clone viral pieces in and out of various vectors, for example. Alright, so another equipment in the lab is this machine here. It's called an FPLC, fast pressure liquid chromatography. What you can do with this unit is basically attach these really high performance columns, and you can bind and purify various proteins to select things from a large mixture of different proteins, right? So you can perform these purifications to get these pure proteins where we'll be able to perform these biochemical assays to see how different proteins interact with each other. So you can use this, purify your proteins, and basically look at protein-protein interactions, simply put.
Mm-hmm. So will thesis students also get to use this machine?
Absolutely. In the various projects that we have going in the lab, we can start looking at the affinities between viral proteins and how they interact with various host proteins. For example, we're very interested in the HIV protein NEF. You can basically purify the viral protein NEF and also purify a membrane trafficking protein such as the adapter protein, AP2. If you have these two pure forms, we can then start to see how these two proteins interact with each other, and we can figure out what are called these protein binding sites, these binding protein interfaces.
Wow, that's really neat. Thanks for your answer.
Thank you very much.
So because you are a professor here at Western, I'm going to ask you a bit about your academic career. So what courses do you teach at Western?
Right, so right now at the undergraduate level, I primarily teach the microbiology of infection and immunity. This is MNI, or Micro M 2500A-B. This course is offered both in the fall and in the winter semesters, as the A or the B, of course. And I am responsible for the virology section. I have been teaching this for many, many years. So this is like a fundamental understanding on the beginnings of virology, but also in this course, other instructors will teach basic introductions to immunology, bacteriology, and of course parasitology. So we try to basically have an introductory piece of all these different infection and disease sections to basically act as a segue into our microbiology and immunology undergraduate program.
Mm-hmm. And I also heard that you took thesis students. So could you describe what a prospective thesis student would be doing in your lab on a day-to-day basis?
Yeah, that's a really exciting part about the undergraduate program and the BMSc program in general. Every year since I've started Western, we'll have between 1 and 3 thesis students in the lab. So essentially these students will work in the lab like this one for a period of 2 semesters. So they'll start in September and they're going to wrap up their honors 4th year thesis around the month of April. So we're looking at roughly, a solid 2 semester experience between 15 and 20 hours a week, roughly, where they're in the lab performing research that, for example, in our lab, will be involved in these viral proteins from HIV and how they interact with host factors. And I'm really proud to say that through the years, a lot of our thesis students have gone on to do fantastic things. They've become graduate students in this lab, they've become graduate students in other labs, they've gone to med school, they've basically fulfilled their dream, right? So I think, doing these 4th year honors thesis within our labs is always a great idea, and it really sets the students on for the future.
Oh, is there one thing that you would tell your students that would help them excel in their classes?
Yes, perhaps to enjoy courses and not to worry right then and there about memorizing and what do I need to know and what do I need to remember. Sometimes just sit back, enjoy the material, and try to understand it a little bit better. I think that's always a better experience for everyone involved.
Yeah, that's really good advice.
Yeah, and to take things day by day. Yeah.
What are the top 3 things you would look for in a prospective thesis student?
Oh yeah, curiosity, determination, and really drive, right? I think those 3 things are just elements that will make you be successful in the sciences and when you're conducting basic science research. There are good days and bad days in research, but the good days are really fun. One of my old mentors used to say it's a roller coaster, right? You go up and down, but it's when you're up, it's really high and it's really exciting.
Sit for a few minutes, right?
Now, I'm sure a lot of students are really curious about what it's like to be a research professor. So what is your favorite part about doing research?
Oh, my favorite part by far is seeing students discover new results and be excited when they see that data. I don't get to have that thrill as much anymore, if not at all. So seeing my students be happy when they get exciting data is the most thrilling part of research by far.
Mm-hmm.
Yeah.
So are there any challenging parts?
There are hard times. We are always at the funding agencies dictate the success and how we run our research program. So that's always something that's I find quite challenging in our profession. But determination again comes into play, and to always remember to always come back and to always fight hard. And things eventually should always fall into place.
Yeah, that's a great mindset to have. And then my final question is, what is your favorite coffee, and when was the last time you broke something in your lab?
My favorite coffee? I really enjoyed espressos. I'm enjoying an espresso, actually an Argentinian espresso right now, the Buenos Aires flavor. It's my favorite from Nespresso. I'm having quite a few a day, maybe probably too much. When did I break something in the lab? I don't know if I broke something in the lab, but I think I've probably broken things more in the lab when I try to fix them sometimes. I always think I can repair something, which is not always true, and I've been told that when I've gone in to fix something, I've made things worse, right? So I try to stay back from these things a little bit more.
Oh, I see. Okay. Well, I think that concludes our interview. So thank you so much again, Dr. Dikeakos, for your time and valuable insight. And I'm sure this will be great for the prospective students who want to go into the micro-M field.
Well, thank you very much for the opportunity. It's been a pleasure, as always. Thank you.